Hello, we will be discussing immunologic tolerance, autoimmunity, and hypersensitivity, presented by Linda Martinez and Heaven Charles. The table of contents include talking about types of tolerance, the factors that contribute to the self-tolerance and immunity, and hypersensitivity reactions. Types of tolerance. Central tolerance is for naive lymphocytes. For T cells, it reacts in the thymus. The naive T cells that are self-reactive will either die through apoptosis, change into regulatory T cells, or are deleted. For B cells, it occurs in the bone marrow, where the self-reactive naive B cells can either edit their receptors through receptor editing or will die through apoptosis. In peripheral tolerance, it occurs in peripheral tissues where mature self-reactive T or B cells can either become inactivated through anergy, die through apoptosis, or be suppressed by Tregs. Molecules that help are the AIR protein, which promotes the expression of peripheral antigens in the thymus to help T cells learn tolerance. And inhibitory receptors are also molecules that can help, like CTLA4 and PD-1, to shut down the T cells. Tolerance to other antigens include commensal microbes, which are gut bacteria that are tolerated through regulatory T cells and have anti-inflammatory cytokines, like IL-10. Fetal antigens are tolerated during pregnancy through Tregs and has poor antigen presentation at the placenta. So, what are the factors that may contribute to the loss of cell tolerance and autoimmunity? First, we have genetic factors, and there are certain MHC genes that are strongly linked to autoimmune diseases. Other non-MHC genes, such as those that regulate immune activation, can also increase risk of things such as self-tolerance or abnormal activation of lymphocytes. Some rare diseases come from a single gene mutation, known as the Mendelian autoimmune disorders. Other factors that trigger autoimmunity are environmental factors. Usually infections can trigger autoimmunity by activating APCs to express more co-stimulators that activate self-reactive T cells, or molecular mimicry, where a microbial antigen looks like self-antigens and the immune system is tricked into attacking the self-tissues. Another factor is the failure of regulatory mechanisms, so like defects in Tregs or inhibitory pathways like CTLA4 and PD-1 can allow for self-reactive cells to be able to escape. Mechanisms of different types of hypersensitivity reactions. So we have 4 types of hypersensitivity, with type 1 being immediate and occurs within minutes, and it's mediated by IgE antibodies and mast cells. Type 2 is antibody-mediated cytotoxic hypersensitivities. They're IgG and IgM mediated and attack cell surface antigens. Type 3 is immune complex hypersensitivity. They're IgG and IgM mediated and they attack free antigens. They're triggered by antibodies that bind to soluble antigens and form complexes that circulate in the blood. Type 4 is the T cell mediated sensitivity, or delayed type. It's mediated by T cells and they react to self-antigens abnormally. So type 1 immediate hypersensitivity has the mechanism of Th2 cells activated and then tell B cells to make IgE antibodies. The IgE antibodies bind to Fc receptors on mast cells. When re-exposed, allergen cross-links the IgE molecules on mast cells, and the mast cell degranulates and releases histamine, prostaglandins, leukotrienes, and cytokines. The clinical effects include the immediate phase that occurs within minutes and causes itching, swelling, redness, other known as allergy. And the late phase occurs within hours later, with more severe inflammation occurring and involves the eosinophils, neutrophils, and T cells. Examples include anaphylaxis, asthma, hay fever, and food allergies. Type 2 antibody-mediated cytotoxic hypersensitivity is triggered when antibodies target specific cells or tissue antigens. The mechanism includes 3 different kinds, with complement activation being one, where the antibodies trigger the complement system, causing inflammation and cell lysis. Opsonization and phagocytosis, where the antibodies will mark the cells to be eaten and destroyed. And abnormal cellular responses, where antibodies will bind to receptors and disrupt normal cell signals without destroying the cells. Clinical effects include local tissue destruction at the site where the antigens are being targeted, and an example is the autoimmune hemolytic anemia. Type 3 immune complex mediated hypersensitivity, like I said, is triggered by antibodies that bind to soluble antigens. The mechanism includes with the larger complexes being able to be cleared, but the smaller antigen antibody complexes do not. When they're deposited into blood vessels, joints, and kidneys, they trigger their complement activation, which leads to immune cells to cause inflammation and tissue damage, like neutrophils. Clinical effects cause the widespread of tissue injury wherever the complexes get stuck, and examples include serum sickness and lupus. And here we have a picture of the butterfly rash that is known as a symptom of lupus. Type 4 T cell mediated hypersensitivity, or the delayed type, is mediated by the T cells, and their mechanisms include with the CD8 cytotoxic T cells. They directly kill the cells that display any of the self-antigens, and the CD4 helper T cells release cytokines that activate macrophages, which cause inflammation and tissue damage. Clinical effects include causing injury and tissue inflammation due to the activated macrophages and T cells. Examples are contact dermatitis, type 1 diabetes, and tuberculosis skin test, and rheumatoid arthritis. And that's a picture of rheumatoid arthritis. And yeah, that is all. Goodbye, guys.